Hello, I'm Rahul Roychoudhuri, and I'm Professor of Cancer Immunology and Immunotherapy at the University of Cambridge and co-lead of the Fundamental Biology of Cancer programme at the CRUK Cambridge Centre. Our lab is interested in understanding how immune cells called T cells recognise, interact with, and can kill cancer cells. T cells are components of what's called the adaptive immune system, the part of the immune system that learns every time you get infected or vaccinated to combat infections when you re-encounter them. Aside from this role, we now know that T cells play a critical role in recognizing and potentially killing cancer cells. We can see this potent ability when we get T cells out of a patient's tumor and can co-culture them in vitro with cancer cells. T cells can often, quite readily, recognize and kill those cancer cells. But when T cells are found within tumors of a patient, their function is potently suppressed. Tumors are Darwinian masses, and as they get killed by T cells, they learn and adapt ways of defending themselves against immune attack. They create a very hostile immunosuppressive microenvironment that prevents immune rejection. Recent breakthroughs in understanding mechanisms of such immune suppression within cancers have led to brand new immunotherapeutic approaches which have really changed the game in the treatment of patients with advanced cancer. These include checkpoint inhibitor therapies and CAR T-cell therapy. But at present, today's immunotherapies only benefit a subset of patients with immunotherapy responsive cancers. These cancers are distinguished by having lots of mutations that the adaptive immune system can recognize.
I'm Alberto. I'm a final year PhD student. In general, what we do in the lab is try to understand the dynamics between the immune system and cancer and how these two systems interact. What I'm interested in more specifically is immune-based cell therapies. So these are quite an exciting development in medicine. There's been a bit of a revolution where there's been the first approvals of cell-based immune therapies. These are essentially a type of approach to cure cancer where a cancer patient gets some immune cells removed from the body. And these immune cells are engineered genetically to train them to find cancer cells and then reinfused back into the patient. And these therapies have worked really well for blood cancers, but they struggle to make success in solid tumors. And what we do here in the lab is try to understand why that is, first of all, and how can we improve these cells so that they work better in the context of solid tumors. So we do things like engineering cells to persist for longer periods of time in the tumor microenvironment, so within the tumor context, which has been a big struggle within the field. But we also like to use synthetic biology and try to give these immune cells new functions that evolution has not provided them with. And so it's a very exciting field because it's very creative and you can really, there are thousands of different ways you could modify cells.
My name is Teresa. I'm a second-year PhD student in the Roychoudhuri Lab, and I'm interested in adoptive T-cell therapies for solid cancers, and in particular in CAR T-cell therapies. So CAR T-cell therapies have not yet shown to be very effective in solid cancers, because as soon as they traffic into the tumor, they face this very highly immunosuppressive tumor microenvironment. So one of the major strategies currently is to armor those CAR T cells by making them overexpress certain cytokines or transcription factors. And in my project, which is in collaboration with the AstraZeneca Partners of Choice program, we are looking to do an in vivo genome-wide CRISPR screen on CAR T cells. So CRISPR is this remarkable tool that allows us to switch genes on and off and then look at what kinds of effects those genetic perturbations have on the fate of that cell. So that will allow us to find genetic means to ultimately render the CAR T cell more robust to this highly immunosuppressive tumor microenvironment and ultimately make them persist longer in the cancer.
I'm Jie Yang. Actually, I'm a postdoc in Rahul's lab in Cambridge. My research focuses on understanding the earliest immune event during cancer metastasis, when cancer cells break away from primary tumor, travel via blood and lymphatic to distant tissues. We found actually, surprisingly, platelets and its blood clotting factor suppress anti-metastatic immunity.
In our lab, we're interested in three fundamental areas. One is in understanding mechanisms of cancer immunosuppression better. By doing so, we hope to extend the scope and efficacy of immunotherapy beyond the existing subset of patients lucky enough to be able to respond to today's immunotherapies to the development of future immunotherapies, which will benefit a broader set of patients. A second interest of our research is borne out by the understanding that some cancers are fundamentally not recognizable enough by the adaptive immune system to be amenable to such checkpoint inhibition, and that we need to create synthetic immune responses, for example, cellular therapy responses or biologic-driven responses to really invoke the power of the immune system against their tumors. Finally, we're interested in understanding how the immune system recognizes and can reject metastases, especially in their early stages. This is a very vulnerable moment in the cancer development story and particularly susceptible to immune attack. And so we'd like to know whether metastasizing cancer cells have distinct vulnerability to immune attack that can form the basis of a completely novel set of immunotherapies with dedicated anti-metastatic activity. We feel that by better understanding how the immune system interacts with cancer, we will be able to deliver better therapies for patients with cancer currently non-responsive to today's immunotherapy.